where is the chromosome? These chromosomes have gone towards one side, these chromosomes have gone towards the other side. So, we cannot draw one nuclear membrane, we will have to show two nuclear membranes. Reason? This is the centriole here, this is the centriole here. Aster rays, they would start to disappear, here they appeared, so they will disappear. These chromosomes which were visible here, now they will again start to change into threads. So here chromatin fiber became chromosome like, here chromosome becomes chromatin like. So it again becomes thin. How many chromosomes are here at each end? 4 have gone here and 4 have reached the other pole. Same thing here, 4 here and 4 here. Now the reappearance of nuclear membrane. Nuclear membrane will reappear around these 4 chromosomes and around these 4 chromosomes. Centriole will remain outside the nuclear membrane. So the nucleus is not 1 now, the nuclei are 2. We said this is nuclear division. Originally we started with 1, now you have 2 nuclei. So karyokinesis has taken place. At the same time, we also start seeing one change. The cytoplasmic constriction also is initiated here. That means the membrane, plasma membrane, starts to show little constriction. We said in the beginning, there is no clear demarcation between one stage and the other stage. So there is a little overlap. Here, telophase and cytokinesis, they are overlapping. So by the time the nuclear membranes reappear, spindle fibers, they dissolve again, aster fibers, they dissolve, the organelle, they distribute evenly themselves. Half of the organelle would come on one side, half on the other side. And same time we start seeing the cytoplasmic constriction also appearing. The next stage after this is telophase. After telophase, what happens is cytokinesis. Nuclear division is complete. Karyokinesis gets completed by telophase. Now cytokinesis. That means the cytoplasm will divide. These constrictions, they will get deeper. Both the halves have their own nuclei now, their own centrioles, and each nucleus has 4, 1, 2, 3, 4 chromatin fibers, 1, 2, 3, 4 chromatin fibers. All other organelles are there, that is Golgi, ER, everything, but we don't draw those things here. Now these constrictions, they would deepen. That means in the next stage, this will get even deeper, this will get even deeper, and we will get two daughter cells with their individual nuclei, with their centrioles, and each one with 1, 2, 3, 4, 1, 2, 3, 4 chromatin fibers. What did we start with? We started with a cell which had 2n number of chromosomes as 4. This is interphase cell, that means it has completed interphase, so it has 4 chromosomes and a copy, because both the daughter cells should get equal number of chromosomes. During prophase, the chromosomes, rather chromatin became chromosome, started condensing, nuclear membrane disappeared, spindle fibers, they appeared, a complete spindle apparatus is formed. In metaphase, all the chromosomes, they align on the equatorial plane. Anaphase, these chromosomes, they start moving apart, rather they are pulled apart by spindle fibers. Telophase is reverse of prophase, nuclear membrane reappears, chromatin formation takes place. Each centriole, it loses its aster, spindle fibers are also lost, and simultaneously cytokinesis also begins, seen in the form of a constriction in the plasma membrane. These two are cytokinesis stages. That means here the cytoplasm splits. These constrictions, they deepen, separating the two cells completely. These two cells which are formed, they are known as daughter cells. Are the daughter cells quantitatively equal or same as the parent cell? Quantitatively means we are talking in terms of chromosomes. 2n number is 4. Here 2n number is 4. So they got the same number of chromosomes as the parent cells. And that is why mitosis is also known as equational division. If this same thing takes place in a plant cell, then how would that be different? In plant cells, there are no centriole. So, no centriole means no aster formation. If no aster formation, then how would the spindle fibers form? Spindles are made up of tubulin. So, those tubulin proteins, which are microtubules, simply they will assemble. So, we will not have these asters, but these fibers would be there. So, chromosomes will get pulled, two daughter nuclei with their nuclear membranes will be formed, and two daughter cells will be formed. But there are going to be some significant differences. So, let us see the differences that take place or that are seen in mitosis of animal cell and plant cell. So, now let us talk about the differences in mitosis that we see in an animal cell and a plant cell. So, in animal cell, the complete process we just now talked of, and in a plant cell, the first major difference is, in case of animal, centriole is present, in case of plant there is no centriole. So, here aster formation takes place. In case of plant cell, there are no asters, and that is why the mitotic division in plant cell is also known as anastral. Reason? There are no asters formed. In case of animal cell, there are two asters formed. So, it is known as amphiastral because of two asters which are formed. Second difference which we see in case of or in between plants and animal cell, in animal cells the spindle fibers, they appear from the centriole. Spindle fibers arise from centriole. In case of plant cell, as there are no centrioles, so there is no question of spindle fibers arising from them. Spindle fibers simply assemble and they arise from ER. So, spindle fibers assemble. So, there is no centriole which is going to give rise to these spindle fibers. Third difference is seen during cytokinesis. In case of animal cells, we said that this cytoplasmic constriction appears or the plasma membrane shows these kind of infoldings. In case of a plant cell, because each plant cell has a rigid cell wall, constriction is not possible. So, a structure appears in the middle. This structure, middle structure, is known as cell plate. These cell plates are formed by fragments of ER and Golgi, and they would secrete middle lamella, and then each cell will start secreting its primary, secondary and tertiary cell walls. So here the constrictions, they appear from outside and go inwards, whereas here the cell plate appears in the middle and it grows outwards. So this division of cytoplasm is known as centripetal. From periphery towards the center, and this is centrifugal, that is from the center to the periphery. One more difference which we see, but it is not a compulsory difference. In case of animal cells, when the two daughter cells separate or they divide, they may separate. In case of plants, these two cells, they will remain together and they will share the same cell wall which is formed in the middle. So these are some differences which we see between an animal cell and a plant cell. So while drawing also, we will have to keep these things in mind. Now let us talk about types of mitosis. Two types we have already talked of just here, the anastral type and amphiastral. So one is anastral, where there is no aster formation, and this is seen in plants or plant cells. The second one is amphiastral, where two asters are formed, which we see in case of animal cells. So here example is animal cells. Third type of mitosis is known as endomitosis. In endomitosis, the spindle fibers, they appear inside the nucleus. So nuclear membrane is not lost. Here spindle is inside the nucleus, and because the spindle fibers are inside, many a times these DNA which have replicated, they don't separate. So this results into polyploidy. Normally the chromosomes should separate, and they separate because these the spindle fibers, they pull them apart. But because everything is happening inside the nucleus, those two DNAs which are formed, one original and its copy, they fail to separate many a times. So those, they remain together. So originally 2N chromosomes, their copies have not separated, so 4N, so it has become tetraploid. So diploid, triploid, tetraploid, hexaploid, so we start seeing ploidies. Fourth type is known as free nuclear or simply nuclear mitosis. In which everything is same. That means it takes place inside the nucleus, spindle fibers are formed inside the nucleus, but karyokinesis is completed. So as a result of complete karyokinesis, many nuclei are formed. So these kind of divisions give rise to multinucleate cells. The basic difference between endomitosis and nuclear mitosis is only one. Here the DNAs which are formed as a result of replication, they remain together. The result is a polyploid cell. Whereas in nuclear mitosis, the nuclear division is complete. That means the DNA which have replicated, they are able to separate, and because of which the number of nuclei would increase. So it would give us a multinucleate cell. So these are the four major types of mitosis that we talk of. There is one more special type of mitosis which is known as dynomitosis. Dinomitosis is reported in dinoflagellates. And in this type of mitosis, it is very much similar to endomitosis, spindle fiber formation and everything, but the nuclear division is complete. So, it is a combination of free nuclear as well as endomitotic division, which is seen in dinoflagellates. So, this is given a special name because it has been reported only in dinoflagellates. So, with this, these 4 types, major and 1 special type, these would be the types of mitosis. What is the significance? What role does mitosis play? How does it help? First and the most important thing is that mitosis is helping in formation of the cells which are similar to the parent cells. So, wherever growth, repair, regeneration has to take place, those cells will divide by mitosis. Because if skin cell is dividing, it has to produce skin cells. If bone cell is to be formed, it has to be a bone cell. So, same type of cell should be formed. Division would be mitotic division. Asexual reproduction. In asexual reproduction, vegetative parts like stem, root or leaf would be helping in division. So, the division which takes place in these cells is also mitotic division. So, one is growth, repair, regeneration. These are the significant points. So, if we have to just write significance of mitosis, first point would be growth, repair, regeneration. All these are due to mitotic division. Second important role is in asexual reproduction. And third is when the zygote is formed as a result of sexual reproduction, the zygote also divides by mitotic division. So, all zygotic divisions are mitotic divisions. That is how all the cells of our body are same as the zygote in quantitative manner. That means the number of chromosomes in the zygote is 46 in case of human beings, 23 received from egg, 23 received from sperm, and all cells of our body are diploid. How are the same type of cells formed? The zygote undergoes all mitotic divisions. So, these are the 3 important points where mitosis is very, very important. There are 2 things. There are certain substances which favor this division, and there are certain substances which inhibit these divisions. So, if a substance favors a substance or a condition, substance or condition which favors mitosis, are known as, those substances or those conditions, they are known as mitogens. Examples, we have talked of phytohormones in the chapter of growth and regulation in plants. Those phytohormones like cytokinins, auxins, they promote cell division. So, they are mitogens. Certain substances, they inhibit mitotic division. So, substances or conditions that inhibit mitosis are known as mitotic poisons. They are called mitotic poisons. Examples of some mitotic poisons, one is cyanide. Cyanide normally blocks the prophase. Then colchicine blocks metaphase. Why it blocks metaphase? Because in metaphase we said those spindle fiber assembly and all those things take place. Colchicine does not let the microtubule assembly take place. So colchicine blocks spindle formation, spindle fiber formation. There are some more substances like carbon monoxide. Carbon monoxide is known for its poisoning effect, which is known as carbon monoxide poisoning. It blocks Krebs cycle, and Krebs cycle is the one which is giving ATP for the complete division. So it is not directly blocking it, but indirectly. So carbon monoxide, it reduces or stops Krebs cycle. And that is how it would affect mitosis, or it would inhibit mitosis. So these are some substances which favor, called mitogens, and some substances which inhibit are known as poisons or mitotic poisons. We have been talking about ATP everywhere, that complete mitotic division requires ATP. Where is this ATP required? ATP is required for almost everything. That is protein synthesis, RNA synthesis, everything. One more important thing where this ATP requirement is very, very high. When the chromosomes are on the equatorial plane and they have to move towards the pole, pulled by the spindle fiber, one chromosome, when it has to move from the equatorial plane to the pole, 30 ATPs are required. So this is very interesting point. 30 ATPs are required to move one chromosome from equator to pole. How many chromosomes are there? In case of human beings, 46 per cell. How many cells? Millions, billions, or maybe trillions of cells. So how much ATP is required? Infinite numbers. So ATP is very, very important, and that's why when Krebs cycle gets blocked, it immediately affects the cell division process. So we have done everything about mitosis, the steps of mitosis, types of mitosis, even significance, and then mitogens and the mitotic poisons. Revise each and every step, go through all the diagrams and just practice it. So this is mitosis. The next division that we are going to take up is meiosis.